Alright, so let's keep going with our little lecture series on physicochemical properties and chemical informatics and computational tools. We're still moving on. We have a little bit more. I added a little bit today in terms of the in silico synthesis. We have a pretty fun little tutorial on that and what it is. It's basically a way we can do organic chemistry inside the computer, and given the fact that we can't be in the organic lab right now, it's probably the best we can do, but we can actually do organic synthesis in the computer in silico. Alright, so let's go through a couple of these topics, and then we're sort of leaning, or we're kind of moving towards a couple of case studies of little research projects I've done in my own lab that use some of these computational tools. But we need to go over a couple fundamentals first. Alright, so what are some of these computational methods in drug discovery? Well, we've done a couple already, the metabolism prediction, and also we did like computational determination of the Lipinski parameters and the Weber parameters. But there's some other tools that are maybe closer to like protein structure and how a drug might interact with the protein. So one of those tools is called virtual screening. What's a definition of virtual screening? There's multiple definitions of this, but this might work for us. Computational technique in drug discovery to search libraries of small molecules. We'd often use something called docking, and it's basically, we can have computer representation of large numbers of molecules that we could maybe dock into a protein, and do this all by computer, and that's why we call it virtual screening. It identifies molecules likely to bind to a drug target. That's definitely our goal, at least. So this is really a computational technique, and we may couple it with experimental drug binding, or like a assay. But there's definitely people that just focus on doing virtual screening. To really be true, I guess, or not make believe, I guess you'd say, people would always follow this up with an experimental assay that to confirm, that maybe a drug activity actually works. Otherwise, it's just really just a computational technique. Okay, so how does this compare with maybe more classical drug discovery? High throughput screening is another tool that we use. It's really experimental, and that's identifying molecules that bind to a drug target, but we actually do this in the laboratory, so we need a real assay, and it's ideally a high throughput screening assay, HTS. And so we're dealing with large number of compounds, massive libraries of purified compounds. In terms of price, which approach do you think is cheaper? Well, virtual screening is basically free, and then high throughput screening, you're buying maybe hundreds of thousands of molecules. You have to purchase them, and you need special robots and a lot of human power. So you can see why people might be interested in virtual screening, because it's definitely a little less expensive. We can do it really easily at SF State, for example. But you usually would follow through with some kind of real experimental screening. Okay, so HTS, or high throughput screening, is an experimental technique. Tons of different assays, of course. That's a huge thing in organic or in medicinal chemistry, is assay development, cell-based assays and enzyme-based assays, and it's all getting into biochemistry and maybe cell biology. Okay, where do we get molecules? If we're considering virtual screening, where would we actually get molecules? There's kind of a couple ways you could do it. Either contact a commercial vendor. So they're a company that actually really sells real molecules, but they may sell you, or they may give you for free the molecules that they sell in a computational format. There's also something called UCSF zinc. You can Google it. Zinc is a depository of about 230 million compounds that are all commercially available from various vendors. And it's convenient because I can go to one website, this UCSF zinc website, and download pretty much 230 million compounds in a format that I could actually do virtual screening with. So that is very, very convenient rather than dealing with multiple vendors. Okay, so some of the vendors though, Cambridge is pretty popular. ChemDiv is another company. Enamine. So these three are pretty popular for small molecules that we could be using for virtual screening or also real screening. You know, these companies, their main function is to sell large numbers of compounds that we might use for high throughput screening. But then we can also use, we can also download their molecule, the sets of molecules they sell in a virtual screening format. And of course now we know SMILES, right? So SMILES is a method where we can to represent chemical structures. And so you can just download from a lot of these companies all their entire catalogs, everything they sell in SMILES format, which is really cool. Sigma Aldrich is another big company. They're maybe more used for like reagents and building blocks of molecules for organic synthesis, but they also have nice collections also for high throughput screening or virtual screening. UCSF Zinc, this is basically this tool that we can download compounds from. And it's pretty nicely designed website, pretty easy to use. You can also search for substructures, like parts of a molecule that might be interesting. In silico synthesis, what does this mean? Synthesize libraries of molecules in the computer. And so we can actually do kind of normal chemical reactions that you're used to doing in a research laboratory, but we can do them in a computer and actually build libraries of molecules. So I could make a library of like 1 million variations of maybe my drug or something. It's a great source of molecular libraries for virtual screening. And then of course what you could do is do in silico synthesis to like identify, design a library, and then you do virtual screening to see like, oh, are any of these molecules maybe good? And if they're really good, then you make them for real in the real laboratory, and then you can actually screen them and have a real experimental result. So all of these techniques can be kind of used in complementary fashion, and that's very very useful for us. Okay. Okay, so computational docking, which I just mentioned a bit ago, is one of the methods we can do virtual screening. So there's other ways to do virtual screening, but docking is a really popular one. Computational technique where a 3D molecule, so we take an organic molecule in 3D format, and we fit it into the 3D structure of a protein, or maybe it could be a DNA or something. Very typically it's a protein. And then we try to identify a minimal energy conformation, which just means that the molecule could be rotated or flipped, or there might be different orientations of it, and so the docking program will try to find the optimal orientation of the molecule where it sits inside the protein. And that's, it kind of emulates a drug receptor interaction. So that's really cool in principle. In practice, it has some hiccups here and there. It's not perfect. Sometimes the energy scoring functions, how we determine the energy, how we quantify the energy of the ligand binding, is not perfect, and sometimes you get false positives. So something might look really great by docking, but then it has no activity at all. So we have to be careful with docking, but it does give us a way to evaluate large numbers of compounds, and maybe narrow it down to a couple that we can either purchase or make in the research lab. That's docking. So where do we get the 3D protein structure from? thinking about what we learned in this course, and also what you've learned elsewhere in your undergraduate education. Where do we get 3D protein structures? So one option is the PDB, the Protein Data Bank. It has published X-ray crystal structures, but not every protein has a PDB structure, right? And so then what do we do? So if there's no X-ray crystal structure, does that mean we can't even attempt to do docking? Fortunately, the answer is no, because there's other ways to kind of estimate the 3D protein structure. Generally, that would be homology modeling. So homology modeling is a useful alternative. It's an estimation of a protein 3D structure using the protein sequence and homologous 3D protein templates. What does that mean? So we basically take the protein sequence, like a protein that we're interested in, and we can, using the protein sequence, there's kind of like a computer algorithm that will look for other proteins that have similar sequences and that do have crystal structure, right? And then using all that, the program will actually kind of generate a possible 3D structure of your protein. So it all depends on there being these templates, right? There have to be templates that kind of look like your protein and have similar sequence homology and have, that resemble your protein. Okay, so it only works well if are good homologous 3D protein templates. Okay, so let's talk a little bit more about virtual screening and docking as a kind of a technique that's used in drug discovery. So this sort of shows how we might take molecules that could either be synthesized, so we might have made some libraries of molecules related to our drug target, or we could do generate virtual screening, like looking at large libraries of molecules to dock against our protein, right? So somehow we come up with molecules we want to dock, and then a structure of the protein might come from homology modeling, so something we might create ourselves, or from structural biology, which basically means from an X-ray crystal structure. So given these, we use this program of called docking. It's a computer program, and some of these are available free. We can actually just download them. Some of them are even web-based. So there's ways to do this, either on your computer or on the web, to do docking. And then what the docking program really does is it fits the 3D representation of your small molecule ligand into maybe a cavity or a place on the protein, and then you get this sort of docked structure. Okay, and then this can be an iterative process. So once you've done this, you can kind of maybe make more molecules or synthesize that molecule, make some analogs of it, and then kind of repeat this process. So it could be a cyclic process, and you can try to improve your molecule and make it fit better and maybe be more active, and then this could generate potential drugs. So for this to really work, you need to also do real biological evaluation. So some kind of assay, whether it's a cell-based assay or an enzyme-based assay, to actually confirm the activity. Because sometimes it'll look really great in the computer, but then it won't have any activity at all. So you need to just use this as a tool, but it doesn't like guarantee that it's an actual real biologically active substance. So some of the homology modeling tools that there are to do homology modeling to generate a protein structure. There's something called Swiss tools that's very, very easy to use, and I'm going to actually demonstrate it on the website. You won't do this in your homework, but you're welcome to play around with it because it's free and very easy. There's something called Yesara, that so Yasara is a paid program. I have it, and I think I spent about $800 on it. It's a program that does homology modeling and docking and a whole bunch of other things all in one computer program. So it's pretty cool, actually. I like it. It's annoying that it costs so much. There's something called Modeler that people seem to like. There's something called Fire. So each of these you can Google to find more information about. But we'll do a quick demo of Swiss tools real quick to show how it works and how we can generate a homology model. So let's try it out. Okay, so what do we need to, if we want to do homology modeling, what do we need to start with? The main thing is you need a protein sequence, right? An amino acid sequence. So I'll do one that for a project I'm interested in that we actually do in my research lab. And this is called, let's see, sorry. Well, what I'm going to do is I need to get the protein sequence. And to do that, I'm going to go to PubMed Protein, which is PubMed is produced by the National Library of Medicine, and they do all sorts of stuff. It's a literature search engine, and they have a lot of different things related to proteins and protein sequences and DNA sequences, X-ray crystal structures, etc. So I'm going to PubMed Protein. And let's, I don't know if you can see it. Whoop. And with PubMed Protein, I'm able to search for a protein sequence. So PubMed Protein, go inside, and I'm going to look for a protein that we use, that we're investigating as a research project. It's called SLC26A3. SLC26A3. It's a chloride channel, so it's a membrane-bound protein, and membrane-bound proteins are notoriously difficult to get X-ray crystal structures on. They just don't crystallize very well, but you can make homology models of them. So SLC26A3, and then when we go there, we can see there's some examples of different species. I don't know what Takufugu obscurus is, or I don't know biology too well. I know musculus is mouse, so Bos taurus is cow. See, these are different species that have this protein. But then if we go down here, we see the chloride channel exchanger Homo sapiens. So this is the human SLC26A3, which is the protein I'm interested in. So I'm going to click it. Okay, and then there's some information about who's kind of discovered it and some of the biological roles of this protein. If you go down to the bottom, we see the protein sequence. So this is the amino acid sequence of the protein, and I can just copy this. But the Swiss tools, the website I'm going to use, it doesn't like it in this format. It likes it in something called FASTA format, which is a slightly different representation of the protein sequence. So I'm going to go to the top, and I can just select that. I'm like, all right, go up here, and you see FASTA. So let's click FASTA. Okay. All right, so now this is the FASTA representation, and it's just, all I do is copy this on the clipboard, and this is the protein sequence. Okay, so now what I'm going to do is I'm going to get on to the web, and I'm going to jump to something called Swiss Tools Homology Modeler. Just Google it. Swiss Tools Homology Modeler. Swiss model. It's called Swiss model. There it is. Okay, and I just say start modeling, and I just paste my FASTA in there. And then I gotta give it a name, so I'm gonna call it Mark, SLC26A3, maybe Mark human. Email address, okay, now we have everything in there. And I think all we do is hit the build model, yeah. And so now that's it, and it creates a PDB file, and the PDB file you can open up in ICN3D or any of your PDB viewer programs. I'm not going to wait for this to go, but it takes about 20 minutes or something. I will show what it looks like though. Let me see. This again is Pymol, which is the kind of cool PDB viewer program that we use kind of in research. It is also it is again available to you. It's a little bit of work to install. But yeah, these are chloride channels, and there's 2 of them. It's a dimer. And so what we believe is that each of these is a chloride channel, and then kind of like there's a membrane domain, which is kind of in the middle. So we envision a membrane right there. And then this is the outside of the cell. It's called the extracellular domain. And then the inside of the cell is at the bottom down here. And anyway, yeah, and so with this we could actually do docking, for example, and we'll show some docking results later related to this protein. Okay, but again, you can also go to ICN 3D, which let me see, ICN 3D, which is the 3D structure viewer, and this is another way to view PDB files. Yeah, I'll open it up. Okay, so let's see. PDB ID. Nope. I need to open a PDB file. So File, Open, PDB file. Browse. Find my file. It's a PDB file. Load it up. And I think I have to do View. Oh, there it is. Yeah, so it is loaded up into this web-based thing that you guys are familiar with. Okay, cool. So that was a demonstration of homology modeling, and we just didn't let it, we didn't wait the 20 minutes it was going to take. Yasara, for the same protein, it takes about 8 hours, I think. It does a lot more work, I guess. But Swiss Tools is pretty cool. It's for something that's free and very easy to use. That's nice. Okay, so then we have some docking programs. And what are some docking programs to do the docking? There's something called Autodoc and Vina, which are related programs, and these are totally free. They're, I think, developed by the Scripps Institute in San Diego. And yeah, Vina is actually known to be pretty easy to use. I think there's some YouTube tutorials on it. And I have colleagues that use it in docking. OE docking and FRED are produced by a company called OpenEye. These are free for academic use, and you basically, well, if you work in a research lab, you can ask to get a license for these. Yasara again was the one I mentioned above. And there's also something called Glide, which I think is produced by Schrödinger, which is a major company that does a lot of computational modeling tools. Okay, so overview of automated homology modeling and kind of what does homology modeling do? And I have my homology modeling experiment started already in Switzerland with the Swiss Tools website. This is basically what it does, and there's a lot to this, but just so it's not completely magic. It takes the protein sequence up top, which is what I copied, the FASTA sequence. What the program does, the homology modeler, what it does is it looks for other proteins that have similar sequences, and then it finds these templates. Templates are like alternate structures of other proteins that are not my protein, but they have X-ray crystal structures. And so it finds these templates. There's a series of these kind of corrective actions, and basically it starts building a three-dimensional model. It first starts with just the alpha helices and all the different parts of the protein, the kind of secondary structure, and then it starts modeling loops, little loops or loop regions that are connecting all the pieces of the protein. So that all of this happens, and it just gradually builds a crazy molecule, a protein structure, a simulated protein structure. And then after it does that, it starts modeling all of the side chains, the amino acid side chains, and the tyrosines and the alanines, and all those things to minimize the energy. So this turns into an energy minimization problem, and then it basically builds a model. It finishes the model, and then there's like a validation step where if there's different clashes or like amino acids are bumping into each other or loops are bumping into each other, there's all these potential problems, and then it the program will fix it. It'll like try something else in the backbone, or it'll try some other loop modeling method. So it's basically an automated process that does this, and the end result is you get a simulated three-dimensional structure of a protein. Super cool. Okay, so molecular dynamics, it's a computer simulation method for analyzing physical movements of atoms and molecules, maybe in a protein, or it could be DNA or something, for an extended amount of time. So when we think about a protein crystal structure, it looks static, right? It's like solid. And what molecular dynamics allows you to do in a chemical or biological sense is to let all the atoms equilibrate and change over time, which is really, really cool. It's a pretty amazing thing you can do. I'll show real examples of this time or next time. That I have some YouTube movies that I'll show you. This is actually kind of a movie thing. It's not just a picture. You actually have to watch the movie, which is really cool. Okay, so it's very slow to do this. It's computationally demanding, so it takes a long time to simulate a protein and the chemical structure of a protein. Docking, like I mentioned, docking can take like 10 minutes. It's a very fast process, but molecular dynamics might take on the order of a week to a month or more to process. So it's kind of a slow thing. This is just an example, random example I found of like how a protein, maybe with a bound ligand, can be simulated by MD, and the idea is that you're kind of looking at the dynamics of how the ligand binds to the protein, and it, through an MD simulation, a molecular dynamics simulation, you can watch, like, does the molecule drift away? Does it stay bound to the protein, or does it just bind temporarily? So it's a dynamic process. And this reference kind of just talks about some of the use of molecular dynamics in drug discovery. I think I have this. I might be able to post it for you guys. Here's just another example. This is a protein called the follicle stimulating hormone receptor, FSHR. And this, I have a paper, PLOS One 2018, and you can download this. PLOS One is it's a open science journal, open access science journal. So everything on here you can freely download. So this is a pretty cool example because it's a very complicated protein. It's one of these GPCRs, a G protein coupled receptor. So what happens is a ligand binds on the outside, the extracellular domain, and then that causes all sorts of changes in the structure of the transmembrane region, and that changes structure, and then there's a G protein cascade in the cytoplasm, which is down here, which causes all sorts of effects inside the cell. So something happens on the outside of the cell, and that causes a propagation through the cell to the inside, and then that causes these G protein cascades. And so what this paper did is they examined all the dynamics of this protein, and it's very complicated because you have the protein, you actually have a biological lipid bilayer. So this is a lipid. It's a lipid bound protein. There's tons of water. All these little red things are water. There's actually water all over the place. They're just not showing it. And there's also, in this case, this protein has cysteines, the sulfur-based amino acids, and they have palmitoyl groups. Palmitoyl is a big, long, greasy acid, carboxylic acid, and so those are attached to cysteine, and that's all the dynamics of this is followed. So they have a pretty good understanding of the structure of this protein and how it embeds into a lipid, and how some of the structural features work. It's kind of hard to model GPCRs, but yeah, this is probably a cool paper to look at. So follicle stimulating hormone receptor, if it has abnormalities or it's maybe underexpressed, it causes infertility in women. So it's an important GPCR in fertility. Okay, so let's see. I want to do a little bit of in silico virtual synthesis. So this is something we can actually do. I have a tutorial for it, and let's try it out. So we're basically going to try to do organic chemistry inside the computer and predict the product of chemical reactions. This is all something you guys can do also, and it'll be a fun thing on the next homework assignment. So, brief demonstration in silico synthesis. What do we mean by this again? It's kind of like making molecules within the computer. So virtual synthesis of organic molecules, or big libraries. We can make big libraries of molecules inside the computer rather than in the laboratory. For this demonstration, we're going to recall from our previous homework the synthesis of potent and selective Notch1 and Notch3 inhibitors. Remember this? So in that homework assignment, you guys looked at this synthesis. It was like this kind of amine component and a carboxylic acid. I think the last step of the synthesis was treatment with triethylamine and HBTU. Triethylamine took off the proton, made a carboxylate. Carboxylate attacked HBTU, and there's a multi-step mechanism. You guys all did this in the homework assignment, so you're all experts at peptide coupling reactions, right, to make amides, remember? So this was actually from the answer key from that problem set. So the base took off the proton and made a carboxylate. Carboxylate attacks HBTU and do some electron juggling, and then kicks off it kind of makes a leaving group on your carboxylic acid derivative, and then the HOBT, which is kind of the other part of this molecule, attacks, kicks this leaving group off, and you get this. And then this is a second leaving group, and then your amine attacks this, and electrons go up, electrons go down, kick it off. And that's how we did this. So you know all about the mechanism. And the big thing about this is that this is an incredibly easy reaction to do. HBTU and EDCI, all of those things are really easy to do in the lab. So amide formation is it's found all the time in drug discovery, and it's easy to do in the laboratory. I mean, it's something that you guys could do in the lab in 5 minutes, maybe. It takes probably longer for you to weigh out the reagents and add the triethylamine or not do all the addition of everything, versus actually doing the actual reaction takes minutes. Okay. All right. So and in this case, this was a potent inhibitor of Notch1 and Notch3, which are anti-cancer drug targets, and so 2 nanomolar, 3 nanomolar. They called it pan-notch, just like pandemic, right? So it's pan-notch means we're inhibiting everything, and whereas pandemic, I guess, would refer to an epidemic over a large population. But in this case, it's inhibiting 2 of these Notch proteins with low nanomolar activity. Okay. All right, so let's show a demonstration in a silico synthesis now. So what we're going to do is consider this reaction, and we're going to look at it from a in silico synthesis perspective. So the building blocks are an amine and a carboxylic acid, right? and they're formed through an amide linkage. And the amide linkage is caused or created, I guess, by the reagent HB2. Okay, and so if we were making a library of molecules, or like, you know, if we're working on this research project to make pan-notch inhibitors, it's possible that one of these sides is not actually absolutely crucial, and that, you know, maybe we could change one side or change the other side, and so that would give me a molecule with either a different left component or a different right component. Okay, and so can we use computers to do this, to maybe change the left part, change the right part, and do large numbers of different molecules? And then maybe we could use like virtual screening or docking to evaluate them. That would be nice. So the bond is created by the reagent HB2. In principle, different building blocks could be used to explore the effect of the building block structure on activity, right? So I could have different variations of the left piece or different variations on the right piece. And, you know, in this case, this is actually a pretty good drug. Well, at least in terms of biological activity. But what we really want to do for you guys is just emphasize or demonstrate the concept of in silico synthesis. That's why I'm kind of using this example. I'm not trying to say that we're actually going to make anything really better than this, but I want to demonstrate this idea. Okay, so we can do in silico synthesis easily and rapidly by a computer. So we can kind of do this reaction or variations of the left or right components, and we can make libraries of molecules that kind of look like the product. And then we combine them with virtual screening or docking. That's one thing we could do, assuming there's a crystal structure. I don't know. I'm not sure about Notch, but if you could do something like that to identify new active inhibitors. And quickly, you know, if I could make a library of like 1 million compounds, and if there's a way to kind of narrow down those 1 million compounds quickly, that would be ideal. We can also, you know, use the automated Lipinski rule of 5 and the Weber rule filtering of a virtual library, and then look for drug-like molecules and maybe focus on the drug-like ones. Okay, right? So the point is that there's a lot of computational tools that we can use after we make a virtual library to kind of evaluate the compounds. All right, so considering this example, one idea is that we could do virtual synthesis using alternate carboxylic acids. So we could keep this amine intact. We could keep this amine intact and then just use a computer program to kind of look at all sorts of different carboxylic acids, right? And then do virtual synthesis with alternate carboxylic acids, and that'll give us a new library where the right side of this is different, right? So here's an example. I can just take this left molecule, and I can, if I use the enamine commercial vendor, I can download, and you know, I actually could. I think I did this today, and I downloaded 15 million carboxylic acids in about 5 minutes. So I obtained 15.7 million carboxylic acids in about, maybe a couple minutes downloading. And then I can do in silico organic synthesis, or we could also call it virtual synthesis, to make 15.7 million products. Okay, and we're going to actually do this in a few minutes, and you're going to do it on your homework also. All right. The other idea is that I could do the same reaction, but keep the right side static, and then vary the left side. So I can take from enamine vendor, I can download 22.2 million primary amines and react it with our carboxylic acid and form the amide bond through in silico organic synthesis to make 22.2 million products. Okay, and I also downloaded these 22.2 million primary amines in about 5 minutes, and they're all smiles, right? So you know smiles. It's a chemical structure representation method that you're familiar with now. Okay, For our demo, we're going to use a subset. Dealing with all the millions of compounds it's just kind of slow on the computer, and we don't really need to do that. So I just made a subset. I just picked some acids from one file. I did about 96, and then the other one I just randomly picked about 1,000. So that was arbitrary. I just kind of picked some from one, some of the other, and in one case is about 100, and the other is about 1,000. All right. Okay. All right, so before we continue, we need to talk about a couple things that we'll just see these. I want to mention them before we go on. Something we know, smiles, right? What's smiles? Smiles is a computer method to represent chemical structure, right? And so we did a whole bunch of lecturing on smiles, and kind of how I can take almost any molecule and figure out the smiles version of it. And we also know about there's basically two kinds of smiles. There's canonical smiles, which is the chemical structure, but in the absence of stereochemistry, so it doesn't look at the wedges and dashes and stuff. But there's also something called isomeric smiles, and isomeric smiles actually does encode the absolute three-dimensionality of these chiral centers. Okay, smarts and smirks. What are those things? Well, let's compare smiles versus smarts versus smirks. So smiles is that's a kitty cat emoji. Smarts would be a cat that is studying for the I don't know, the GREs or something. And then a smirk here is a smirk. But in terms of chemistry and chemical informatics, these actually mean something different than these silly emojis. Just like smiles, as we know, refers to chemical structure. Okay, so smiles is a computer representation of organic structure, and so for these two molecules, I gave us some smile strings, and these are isomeric smiles, as we see by the @ sign. That the @ sign indicates a chiral center, so that's probably the chiral center right there. And then here's another smile string for the other starting material. So this one is for the left molecule, this one's for the right molecule. Okay. And then we have smarts. So smarts is a computer representation of an organic substructure. It's a fragment or functional group. So that's what smarts is. Smarts is not like the full molecule. It's kind of like smiles, except that it's only for a piece of a molecule, like a fragment. And so, for example, like when we think about this reaction we're doing up here, what are the relevant functional groups? Well, we have the amine, this primary amine, and we have the carboxylic acid, right? So what we can do is find a smarts string for the amine or the carboxylic acid. Down here is it's a URL, daylight.com, blah blah theory, the word theory, and this is all in the full PowerPoint that I'm putting on iLearn. This company is, I think, responsible for the definition of smiles and smarts, and so it's a good resource if you ever would need or have questions about smiles or smarts. Well, anyway, smarts is a fragment or like a functional group. So when we think about amines and carboxylic acids, well, they have a smarts representation. So an amine, this is the weird computer representation of an amine, and this is the weird computer representation of a carboxylic acid using smarts. Okay, so these and like if you go to this website, this daylight.com, they have like 100 different functional groups, and they have the smarts designations for each of them. For our example, though, amine and carboxylic acid are the ones that we care about when we're making an amide linkage. Okay, so then what is smirks? So smirks is going to be kind of like smarts, but to indicate a chemical reaction. So smirks is a computer representation of a chemical reaction. So it's going to be, in terms of a chemical reaction, what it's going to do is kind of look at you know, the smarts of an amine, the smarts of a carboxylic acid, and then kind of the smarts of an amide linkage. And then it basically, for amide formation, it's this crazy long thing. So this is the SMERCS for amide formation reaction. So this string, it looks like crazy stuff. This defines a chemical reaction between an amine and a carboxylic acid to make a amide. Okay. Some of it kind of makes sense. I don't know if you look closely. This part is basically, it looks like the carboxylic acid part, C double bond O OH, right? C double bond O OH. This is an amine part, NH2. Okay, and then here, this is kind of like the product. So this sort of shows the formation of the product. It doesn't totally look like the smarts, but it's basically what people say is SMERCS is the same as, well, it's smarts applied to a chemical reaction. But there's some slight differences between all of this and these two guys. Okay? All right, so now we know about SMILES, smarts, and SMERCS. And the other thing is, like, for almost any reaction you can imagine, there's a SMERCS string for it. So the aldol condensation has a SMERCS. And so this would be a organic reaction that maps a starting, maybe two starting materials to a product, right? And so there's like kind of websites where you can just get a SMERCS for different chemical reactions. All right, so finally what we've been waiting for, a demonstration of in silico virtual synthesis. Let's try it out now and see if this works. So what we're going to do is we're going to look at this reaction again. And we're going to first, there's the SMILES, the two SMILES strings for the starting materials. And we're going to use this thing called the RD-Kit reaction enumerator. It's a web-based tool at UC Irvine, and we actually used a UC Irvine tool previously. Yes, and it's the Anteater, UC Irvine Anteater is the mascot. Okay, so what we're going to do is go to this website, and this is where the tool resides, and then we're going to click the button for the reaction processor. Before I do that, the other thing is we need the amines and the carboxylic acids, and I have those on my website. So this is a Plasmodium SFSU EDU ~mark chem477. Okay, and so we're going to use all this. The first thing we're going to do is a boring reproduction of this reaction. We're just going to basically use this tool at UC Irvine, the reaction processor, and we're going to just see, can we carry out this chemical reaction within the computer. Let's try it out. Okay, so the things I'm going to need is I'm going to need this SMILES, basically just the top SMILES string and the bottom SMILES string. Okay, and I'm going to have to go to this website. So let's do all this, and I'll do it on my browser for you guys. Okay, so first thing, I'm going to go to the UC Irvine website. All right, so here we are. We're at the UC Irvine ChemDB chemo informatics portal. Okay, and then we're going to click this thing called the reaction processor, which is over on the right. Reaction processor. Generate product libraries. Okay. All right, so what do we got to do here? So the top part is the kind of like the starting materials of a reaction we're going to do. In our case, we have two starting materials. We're going to have these two, the starting materials of the reaction, and so what I'm going to do, these are some examples. I'm just going to delete them. So delete the old ones, these reactants. All right, I can delete these, and then I'm going to get the SMILES of the amine. Whoops. Okay, here we are. So I got the smiles of the amine, and I'm going to paste that. Whoops, one sec. Okay, there they are again. All right, so let's go back. Keep going. All right, sorry. Okay, sorry. Here we are. Okay, so we're going to do this boring reproduction of the above chemical reaction. We went to the website, we hit reaction processor, and what I did is I pasted in the first smiles, the amine, okay? I got it from my PowerPoint. Remember that in addition to these videos, all of my PowerPoints are on the iLearn site, so you can download them, and you should be able to open and like copy these sort of PDF, these smile strings right out of the PDF file, okay? All right, so I pasted the first one, which is the amine, and now I'm going to paste the second one, which is the carboxylic acid. Okay, so let me go get that. Okay, so now you can see they match. I have the left one, amine, and the carboxylic acid, and there they are. All right, so let's look at this a little more now. I'm not going to really change any of this, but here are the smirks. Remember the smirks? Well, it has a whole bunch of default reactions, and the reaction names are on the right. So we see like ester formation, Diels-Alder reaction, internal nucleophilic acylation, and SN2 reactions, aldol, enolate reactions. All I'm going to do is I'm going to get this one, the amide one. That's the one we want, amide formation, okay? I'm going to cut it and put it onto my clipboard, Control X on my PC, Control X, it's on the clipboard. Now I'm going to delete everything else, delete it all, and paste. Okay, so now we have just amide formation. Okay, so what this is going to do is it's going to convert these two pieces, and it's going to build an amide using the smirks. Okay, anything else to set? I'm going to turn off include reactants. Wait a sec. Yeah, turn off include reactants. And I'm going to press the generate products. And this should carry out the chemical reaction. Ah, it did. And so it actually created a smiles string of the product. So it took smiles number 1 and smiles number 2 and created the product. And we know what it's supposed to look like. It should look like this, right? It should look like the product, right? Let's see if it was because we can actually view it down here and see view structures. Ah, look at that! It matches. So yes, it looks like our in silico synthesis worked, and we actually synthesized our molecule in the computer. Okay. All right. So let's move this out. All right. And that was the boring reproduction of the above chemical reaction. Not that exciting because all we're doing is what we know should work. So now let's do something more exciting. And we're going to attach the above amine with 96 carboxylic acids. So we're going to take the amine part, the top part, and then rather than react it with this carboxylic acid, we're going to get 96 carboxylic acids from Enamine, the company. I already did this and I put them onto my website. So let's take a look at my website. So I'm gonna put this on the clipboard. Plasmodium SFSU edu mark chem477. Okay, so here's my website. Well, this is a page on my website. Okay, so here's for my Plasmodium SFSU edu tilde mark chem477. And so what we're going to do is we want to get these 96 carboxylic acids. So that is right here in this file called enamine carboxylic acid subset filtered. Okay, click it, and now you see all these smiles of these 96 compounds. I'm just going to copy the whole thing. Control A to select all. Control C puts it on the clipboard. Okay. Go back to the Irvine utility, and it still shows the previous reaction. I can press the X button. Okay, and now it looks like it did before. We have the amine on top. Yep, the amine is on top, and the carboxylic acid is on the bottom. I'm going to delete the carboxylic acid. Okay, so now we just have the amine on there. Now I'm going to paste all of those 96 things I got off my website. Paste, Control V. And so now I have the amine on the very top, and then all the carboxylic acids. And this website now is just going to do 96 organic reactions when I click Generate Products. I recommend the include reactants. We turn that off over here. Okay. All right. So let's do it. Generate products. Let's see if it does it. It says going, going, going. Oh, there it did. So I just made 96 chemical reactions with those carboxylic acids, and I can actually look at them. And what are they going to look like? They're going to look like this amine coupled to different carboxylic acids. Okay. So view structures. Okay, so there we go. We see the amine part, and then these are commercially available carboxylic acids that enamine sells. And now they're part of our virtual library. We just created a virtual library of 96 different molecules based on commercially available fragments. Okay, so each of them you see this kind of part that was in the original molecule. All right, and we just built a library. Okay. All right, so and they have various structures. They often have a water. I think they all have a water. And I think when the reaction occurred, when the amide formation occurs, you're losing a molecule of water, and it's just showing water being produced as a side product, even though, of course, we don't care about that for our drug. But yeah, that was an example of a virtual synthesis. All right, so let's hit X again. Now we're back to where we were. Now we want to do the other. Let's move this out of the way. Okay, so that was attach the above amine with 96 carboxylic acids. And what could we do with these again? Let's just look at it again. What could we do with these 96 molecules we just synthesized? Well, one big thing is if we have a protein structure of Notch, the drug target, we could dock them. We could dock these virtually synthesized molecules and actually kind of discover new analogs that we could screen for activity. All right. Another trick, of course, is that we could, rather than just use random carboxylic acids that just I just picked 96 random carboxylic acids. We could alternatively pick 96 carboxylic acids that kind of resemble this structure, right? So they might be sort of an amino acid type derivative, or have 2 carbonyls, or different R groups like this. So they might be a little closer resembling of this, and that might be a more meaningful study. But right now I'm just trying to show you that, yeah, we can actually make molecules in the computer for virtual screening purposes. Okay, that was demo number 2. Attach the above amine with 96 carboxylic acids. So now what we're going to do is attach this carboxylic acid with 1,000 different amines that are all available. Okay, so we're going to do it the same exact way. This time I gotta get this smile string of the carboxylic acid, which is the second one. So let me get that real quick on my clipboard. Okay, so I have this on my clipboard, the carboxylic acid, and then we're going to go back to the website. Okay, this is the UC Irvine website. Click X, we're back in it. delete everything from the top. Control A, delete. It's all gone. Delete the starting materials. We're still keeping the amide formation smirks inside there. Okay, so now I paste the carboxylic acid, and you see like this CF3 fluorines. That must be these fluorine. There's 2 CF3s, yeah. So this looks like the carboxylic acid based on the fluorines and stuff, right? Okay, so we just put this molecule in, and now let's go to my website and get the amines, because we're going to attach the carboxylic acid with 1121 amines, right? Okay, so let's go to my website. All right, and now we're on my website, and now we're going to click enamine amines substrate filters, and we're going to click the top one, and now these are the 1,000 amines as smiles. Copy them. And I'm just going to paste them into, not the smirks, but the reagents, which is smiles. So paste those 1,000 molecules in. And now we're going to do the virtual synthesis by clicking Generate Products. There it goes. So we just built a virtual library of basically 1,100 analogs, where the right side of the molecule, the carboxylic acid, is attached to 1,100 different amines that are all commercially available. So we have a virtual library. Now this should work. So we go down. These are all the smiles strings of the product, right? These are 1,100 smile strings of new products we just created. And then to look at it, we just go to View Structures, and then we can actually look at these. Okay, so we can now see resemblance to our molecule, right? It has the carboxylic acid with the tert-butyl ester, so that's kind of the right side of the molecule. And then these are amines, enamine cells that this program attached. So we can go through some of these, and they look pretty drastically different than the original drug. But that's often what you're trying to do, is find diverse chemical structures, right? So they all have this kind of right side of the molecule, and then these different weird amines. And if we let's go deeper in, and they'll be totally different. All these diverse molecules. So, yeah. So maybe one more thing we'll do real quick is. Okay, so we did the demo 3, attach above carboxylic acid with 1,100 amines. We can't this second, or probably easily, evaluate these molecules for Notch inhibition, right? We would need to have an assay, and we'd have to probably make them. But the one thing we can do is we can look at their drug likeness, because that we do have an evaluator for. We can do the Lipinski parameters and stuff. So let's as a last demo, take one of the synthesized products and examine the Lipinski properties and Weber properties, because we can do this. We don't have a good way to virtually screen this library right now, but we can at least do Lipinski. So how do we do that? Let's just close this. All right, and what I'm going to do is go to this box that says Generated Products, Smiles. You can't. I'm just going to get the first one, but I can't get the overall line has a little bit of junk at the end. It says smirks, reactants, blah blah blah. We don't want that. So all I'm doing is getting the smiles. So I'm just going to copy from here to here. I'm going to copy just the smiles. Okay, and if that's a product, an organic molecule we just made. And what I'm going to do is use the Mole Inspiration website, which we used before for Lipinski and Weber criteria. And let's do that real quick. All right, so let's go to mole inspiration. Google it. There it is, mole inspiration chemical informatics. And I think we use the molecular properties, calculation of molecular properties. Yep. Okay, and we got the SMILES string. This is one of the ones we just did in silico synthesis to generate, and you calculate properties. And so this one actually, if we look at the Lipinski rule of 5, which we talked about multiple times before, hydrogen bond donors less than or equal to 5, hydrogen bond acceptors less than or equal to 10, molecular weight less than 500, C log P less than or equal to 5, the partition coefficient. And well, if we look at these things, it looks like our mi log P, which is the mole inspiration version of log P, well, it is greater than 5. It's around 7. So this is probably, that's not a good sign. The molecular weight is above 500, so that's a violation. Hydrogen bond donors and acceptors don't seem to be a problem. But we have 2 violations of Lipinski. The molecular weight and the C log P, or C log P or mi log P. Okay, so that's probably a bad sign about this drug being, you know, a orally available drug. And then in terms of Weber rules, we'll just use their rotatable bond count, and it should be less than or equal to 10. Well, it's 19, so that's a bad sign. And the polar surface area, PSA, is 98, so that's actually not that bad. But overall, this is probably a bad molecule. So we were able to do this, you know, determine this by computer. Anyway, so hopefully this is cool, useful to you if you've never seen this before. The fact that we can do basically organic chemical reactions within a computer and make virtual libraries. To be really cool, you know, and the way people use this in drug discovery, you would need a docking or a protein crystal structure or something that you can dock against. I will show an example of that as we proceed, and I have a couple examples from my research lab, and they're in this PowerPoint series. So we'll see those. I think that should be definitely next time. Okay, all right. Have a great night.